Hello everyone, welcome to Dr. K. PREM-PRIMER Biotech Lectures. In today's lecture, we will talk about TA plasmid features. As you all know that Agrobacterium contains TA plasmid. T stands for tumor, I stands for induction. So have, once the TA plasmid's TDNA transferred to the eukaryotic plant cell, it induces the expression of the genes which are transferred, and for subsequently the plant cell will develop a crown gall disease. So in this lecture we'll see what are the different elements of TA plasmid and their functions in development of crown gall disease. So this is a basic structure of a TA plasmid. And try to subscribe my channel. That's Dr. K. PREM-PRIMER Biotech Lecture for more updates. Yeah, here you can see this is the TI plasmid. So the T stands for tumor, I stands for tumor inducing plasmid. That is around 140 to 200 kb in size. So the various species of Agrobacterium have a different size TI plasmids. Those TI plasmids are responsible for tumor induction and followed by the crown gall disease. You can see the statement here. Most of the genes involved in crown gall disease are not born on the chromosome of Agrobacterium tumefaciens, but on a large plasmid termed TI, or tumor inducing plasmid. The size of the TI plasmid is generally large, ranging from approximately 140 kb to over 200 kb. So, crown gall disease is completely attributed to the TI plasmid of Agrobacterium tumefaciens. And the size of that is 140 to 200 kb. So, we will see the important features of TI plasmid and their subsequent functions in development of crown gall disease. Here you see this is what is called as TDNA in TA plasmid. The TDNA is present between the left border and the right border repeat. These are the 24 repeats, 24 DNA sequences which are conserved among the TI plasmids. So in between the right border and the left border, there is a TDNA region. This TDNA region consists of genes responsible for synthesis of phytohormones such as auxins, cytokinins. So auxins and cytokinins secretion, will help in the cells to undergo the continuous division of cell or differentiation of the cells into a masses. So this is what TDNA. In addition to these two auxins and cytokinin, there are genes which are responsible for opin synthesis also. Opins are unusual amino acids. Those amino acids serve as a nitrogen as well as carbon resource for for a for in case in plants. Then coming to the second one is left border and right border. Right border, you see this is a right border, and the and oh sorry, here I'm talking about open synthesis and open catabolism. Both synthesis and require genes also there to catabolize those unusual amino acids or sugar molecules. And third important element is right border and left border of the TDNA region. So these are repeats are in a 24 nucleotides size, and they are very much conserved. So during the transfer of TDNA, a single strand nick is created by the VIR-D gene here and here. This is a pi prime and this is a 3 prime. So pi prime right border of the single strand TDNA is very important. So uh, the molecule which is responsible for VIR-D2, which is having a nuclear localization signal, will binds to here, and it will help in the transfer of TDNA from from bacteria to the plant cell. So if you create a mutations in the right border and left border, nicking will happen in the left border. It is not possible to have a nick in the right border. Hence, so transfer of TDNA from TI plasmid into the eukaryotic cell, right border is very essential. The next one is origin of replication. Here, this is the origin of replication. You can see this is the origin of replication. This origin of replication will help the cell, will help the plasmid to undergo autonomous replication in the Agrobacterium tumefaciens. The other one is that fifth one point core element is TRA or conjugal plasmid transfer. So this will help in the transfer of TDNA from bacteria to the bacteria to other bacteria. So then you have other one is called as INS. This is an incompatibility region. This is helps in the incompatibility of the plasmids. And the seventh one is a most important one. That's a VIR genes. So region from ear to ear is encoded by the VIR genes. These are the actual proteins which are helping the identification of signal and followed by the nicking of the T plasmid, tDNA, and transferred to the transferred to the plant cell. So you can see vir A, vir B, vir C, vir G, vir C, vir D, vir E, vir F, and vir H. So here the vir A is very much responsible for identification of wound or injury in the plant cell. This is acts as a receptor, and Vir-G will bind to the Vir-A and activated by the Vir-A, and Vir-G is translocated to the nuclei translocated and upregulate the other Vir genes. And Vir-D is an endonuclease which cuts at the left border and right border, nicks at the left and right. And Vir is a single strand binding protein. Once this single strand of TDNA comes into the cytoplasm, it will the Vir E proteins are bounded to the single strand, and the Vir H and Vir F are having the different functions. So the same thing we can see in the text format. See, Agrobacterium transfer TDNA, which makes up a small, approximately 5 to 10% region reside of the TA plasmid. As I told you that the tDNAs which is responsible for tumor induction sequence homologs to this region are always transferred to the plant nuclear genome. As you know that tDNA have enzymes which are responsible for the phytohormone synthesis, such as auxins and cytokinins. In addition to the phytohormones, it also have a site for opin synthesis, which encodes unusual amino acids and sugar molecules, which are collectively called as opins. They are serves as the nitrogen and carbon sources. The tDNA is either side. There are two repeats, left border and right border, whose sequence length is 24 base pairs, and they're very important in the nicking and the transfer of that tDNA into the plant cell. Origin of replication helps the plasmid to undergo the autonomous replication, and Tra helps in the conjugation, and INC region is helps in the incompatibility, and the last one is uh, virgins, whose function is very essential in the development of the pathogenicity and transfer of the single strand DNA from bacteria to the plant cell. So you can see the TDNA. The virgins region consists of approximately 10 operands. So in nutshell, there are seven important elements which are very essential for each and every function, and especially virgins are important for the transfer of TDNA from bacteria to the plant cell, and TDNA containing regions are involved in the development of the cell, crown gall disease. And open genes are which are encodes the unusual amino acid and serves them as a carbon and nitrogen source. And as you all know that origin of replication and Traj regions are responsible for replication and conjugation. So this is all about the various elements which are present in the TDNA, TI plasmid and their functions. So I think hope you understand. I'm thinking that you all understand the components of the TI plasmid and their respective functions. So with this, I am closing my today's lecture. That's TI plasmid general features. Thank you all for listening. If at all you like it, try to give a thumbs up. And if you want to be updated with my channel, try to subscribe. Thank you all.